Welcome to another Global Immunotalk. In case it's the first time you're joining, my name is Elena Zúñiga from the University of California, San Diego, and I'm delighted to be here connected with our global immuno speaker today, Dr. David Brooks from Toronto. And so before I introduce David, as always, I want to remind everyone that our next global immuno speaker is Dr. Marco Binolo. So we hope that you can join us next week as well. And also remind you that in these formats of global immunotalks, we do asynchronous Q&A via Twitter, so people from a different time zones or that couldn't connect live can also participate in the discussion. And for that, you have to search in the account Global Immunotalks for a tweet that I will upload in a few minutes that says, ask questions for Dr. David Brooks here, and reply to that tweet with your question and mentioning the hashtag Global Immuno, and David will answer the questions at his availability. And so without further ado, I would love to introduce to you Dr. David Brooks. It's actually my pleasure and delight because he's a very good friend. So David is currently senior scientist at Princess Margaret Cancer Center and a professor at the Department of Immunology at the Faculty of Medicine at the University of Toronto in Canada. So I should stop sharing screen. There you go. So David is an LA boy. He was born and raised in Venice, California. He performed his undergraduate studies at the University of Arizona. And in 1997, he came back to LA to perform his PhD at the University of California, Los Angeles, at the Department of Microbiology and Molecular Genetics, having as a mentor Dr. Jerome Zak. And there he studied the generation and targeting of latent HIV infection. And probably there is where he developed this lab for chronic viral infections and eradicating chronic viruses. In 2003, he moved to San Diego, La Jolla, California, to perform his postdoctoral research training at the Scripps Research Institute with Dr. Michael Alston. And there is where I overlap with David, and we were colleagues together at Michael's lab. And there he did amazing work where he studied the CD4 T cells and IL-10 in viral persistence. In 2008, David moved as an assistant professor tenure track at the Department of Microbiology and Immunology at the University of California in Los Angeles again. And he was, in 2014, promoted to associate professor with tenure at this institution. But in 2015, he decided to leave California and he moved to the University of Toronto as an associate professor in the Department of Immunology in the Faculty of Medicine, and also as a senior scientist at the Princess Margaret Cancer Center. And now he's professor at the same department. So David Labs is focused on understanding the molecular mechanisms of cellular interactions that promote immunosuppression to inhibit immune responses. His goal is to use this information to devise therapies that block these suppressive pathways to broadly restore immunity to eliminate cancer and chronic infections. So his laboratory was the first to discover many new mechanisms of immune regulation during chronic infections, and many of these discoveries are being tested in preclinical and clinical studies to treat human diseases. So he really had made a number of seminal contributions to the understanding of how immunosuppression develops in the context of a chronic infection. He was, for example, the first to identify the critical role of IL-10 to inhibit antiviral immunity against chronic viral infections and have extensively described the mechanism of action of IL-10 and other key suppressive factors such as Pdl1 and PD1. He has also looked at the regulatory mechanisms that lead to the expression of these factors and how they specifically suppress the immune response to promote disease. Another example of David's amazing science is his discovery of IL-21 as a key cytokine that is produced for CD4 T cells to promote survival and accumulation of numbers of virus-specific CD8 T cells in the context of a chronic infection. So putting the focus on IL-21 rather than IL-2 in the CD4 help to CD8 T cells in chronic infection. And just to name another example, David's group has also discovered that in addition to their critical and well-established antiviral and immunomodulatory roles, type 1 interferons are co-regulators of the diverse suppressive pathways that inhibit immunity during persistent viral infection, and demonstrated that type 1 interferon signaling could be blocked to restore immune health and control chronic viral infections, which I think that we will all agree that was a quite unexpected finding. David has published these important research contributions in top-tier journals and has received many awards for his impactful work throughout his career, and for example, the AARI Early Career Faculty Award. On a personal note, having spent about 5 years working in the same lab as postdoctoral fellows with David, I can say without hesitation that David is a terrific colleague and friend. He is a really nice person to have around, super optimistic, super cheerful and positive, and lots of fun to discuss science with. So I feel lucky to have that life has crossed our paths and we had time to spend together in Michael Alston's laboratory. So without further ado, I would like to mention the title of David's talk, which is Acquired T-Cell Dysfunction and Restoration in Cancer and Chronic Viral Infection. And thanking David for all the efforts to present here, for accepting our invitation. It's very nice to host you, David, today.
Thank you. It was thank you so much for the invitation. It's great to be a part of this series. I appreciate it very much. And the introduction. I almost don't even know who you're talking about. I'll have to look up this David Brooks character.
Okay, so Dave, before I ask you to share the screen, we want to know more about you and inspire a young immunologist. And so we have prepared a question for you. That is, what is the trait in your personality that helped you the most in your scientific career?
You know, so I think the most important trait is probably perseverance, but I think what has made that possible is picking a kind of a research topic and a field and a topic within that field that really interests you. I find that one's going to go through a lot of difficulty and a lot of rejection in this field, but that if you're really working on something that interests you, all that stuff is relatively easy to get through because you just really want to know the answer. So picking something that really just enthralls you, I think, is the key to success and ultimately the ability to persevere in this field and have fun.
Very nice, Dave. I couldn't agree more, right? Like it gives you the drive to overcome the difficulties. I agree 100%. Yeah. Okay, thank you so much. So it's time to share your screen.
All right, let's do this. Last time or a second ago, I think there were a couple things. So let me work through this. So what do we see here?
So I see the PowerPoint, the with the little slides on the left, like the regular PowerPoint. You need to project the slides.
Yeah, let me try to do that. It seems to be the way we had it worked out a second ago. That little thing isn't popping up anymore.
Okay.
So let's see. Okay, I think we're in the home stretch here.
Let's see. Okay.
Are we good?
No, this is still not. Oh, there it is. Perfect, Dave.
Okay.
Excellent. You can start.
All right. Thank you very much. Thanks, Selena. And thanks everybody for attending. It's a real honor to be speaking at this series, and I appreciate it. So I'm going to talk today about some of our lab's work, some of it published, some of it unpublished, really in understanding how T cells function and can be restored, and the role of the inflammatory and suppressive environment in regulating T cell responses during chronic viral infection and then in cancer. And so by way of introduction for us, our lab is really focused on trying to understand immunity to chronic disease and how the inflammatory environment and the suppressive environment can both coexist and actually regulate each other, and then how these can affect immune responses, T cell responses, and ultimately disease pathogenesis. So we have models in our lab, which are three main research areas. We're interested in chronic virus infections, and this is mostly done with a lymphocytic choriomeningitis virus, or LCMV, model of chronic viral infection. We actually have a part in our lab that's really interested in understanding Mycobacteria tuberculosis and the immune response to that, and a part that's really looking at how anti-cancer immune responses are mounted and how they can be restored to try and overcome cancer. And, you know, I present these as if there are three pillars, but the truth of the matter is we're actually working very much in collaboration across lab, trying to understand kind of the shared and divergent mechanisms of inflammation, immunosuppression that really lead to kind of a same T cell or immune dysfunction across these diseases, and understand similarities and differences with the high hope of perhaps finding something that can be used universally to treat these kind of chronic diseases. But for this talk, I'm going to focus mostly on chronic virus infection and cancer, although we published a paper earlier this year on how, here, let me get my pointer, on how chronic virus infection affects the immune response and ultimate course of TB infection. And if you're interested in that, there's, I think, a wonderful study published by Wenzhou in our lab, and you can reference that. But for this talk, I wanted to focus really on two kind of vignettes in the lab that are currently ongoing, and that really have been a foundation in our lab, trying to understand the role of type 1 interferons in these chronic infections, how kind of the choices, how these interferons, so interferon alpha and beta, can have really diverse effects across the immune system, and how we've taken this from chronic infection into trying to interrogate some of this stuff in cancer. And then second, in trying to understand what really are CD4 T cells, and what is CD4 T cell help in infection and in response to immune restorative therapies. So just kind of so everybody's on the same page, at least for how I'm seeing this, in a very simplified explanation, of course, is kind of how is an immune response mounted, in this case, in response to a viral infection. So in response to this infection, dendritic cells or myeloid cells will take up antigen, will present that to T cells. You'll get a T cell priming, T cell amplification. You'll get a real global immune activation of all aspects of the immune response. You get a mounting inflammatory environment, often pro-inflammatory, cytokines, interferon gamma, TNF alpha, things like this. These then lead to an amplification of the T cell response, both CD4 and CD8 T cells. These T cells acquire very strong antiviral cytolytic activity if you're a CD8 T cell, the ability to guide immune responses if you're a CD4 T cell to stimulate B cells. These then, the vast amount of times, culminate in a resolution of virus infection, and then ultimately the generation of a memory response that's able to rapidly respond if that pathogen is encountered again. And this is really, I think, the ideal situation. This is what's hoped for with vaccination and in any immune response. However, in some instances, this pro-inflammatory environment can actually turn into more of a building anti-inflammatory environment characterized by proteins, suppressive proteins such as IL-10, Pd1, Pdl1, CTLA4. There's a whole variety of these that work in concert to ultimately attenuate T cell responses, this T cell exhaustion, as it's been termed, which are really an attenuated response, an inability to produce cytokines, of T cells to produce cytokines to kill infected cells or tumor cells as they might be under an acute condition. This ultimately leads to the inability to resolve this infection. And these cells then, these exhausted T cells contract, but still there and exist in this kind of homeostasis where they still exert pressure. They're still controlling virus replication, but they're incapable of ultimately eliminating that infection. And so we use, in order to kind of understand this process and model these responses, we very often use two strains of LCMV. So the LCMV Armstrong strain, which generates this acute infection, cleared by the immune response, and LCMV clone 13, which due to 3 mutations is able to outcompete the immune response, both replicate faster, infect cells with, and get better infect cells, ultimately outcompetes the immune response, leading to this exhaustion, immune exhaustion. And importantly, using this model has really uncovered many immune mechanisms of how these cells exist in this chronic inflammatory, chronic activation-induced state, and have been then translated into many human chronic infections as well as cancers and other chronic diseases. So it's really served as the prototypic model to understand how immune responses are generated and then maintained in these differing environments. And so in these chronic infections, we talk about T cells, CD4 T cells, you have some skewing of these responses, CD8 T cells, which exhaust and have this attenuated function, and ultimately these CD8 T cells are usually the target of many of these immune restorative therapies. However, the truth of the matter is there are alterations in every level of the immune response, alterations in the innate immune response, everything from dendritic cells and macrophages. There's changes in the fibroblasts and the stromal cells, B cell dysfunctions, as well as changes in either infected cells or tumor cells that all together culminate in this immune exhaustion and inability to either control infection or eliminate a cancer. And the truth of the matter is we study these because one really has to in these very kind of boxed ways, but in reality, these really all exist. And every one of these different perturbations has an effect on every other aspect of the system. It's very much like the butterfly effect, in that a small change in this can have a huge impact on the rest of the immune response. So, we try to really think of a lot of these things in terms of not just targeting one specific aspect, but really how are these holistically being modulated. And again, we build this as these dysfunctions, but the truth of the matter also is that it's very much a balance during these chronic inflammatory states or these chronic infections of both positive regulation, where you have factors, as Alina mentioned, IL-21 or other factors that are sustaining the immune response. In the absence of these things, things can go very, very bad for the immune response. But you also have factors on the other side that are actively inhibiting this immune response. And really, it's this balance that contributes to this whole dynamic of immune exhaustion and immune function during these viral infections and cancers. And one of the observations we made some time ago was that, in fact, type 1 interferons, so interferon alpha, interferon beta, are heavily responsible for modulating both the inflammatory as well as the negative regulatory aspects of these infections. And we became really interested in trying to figure out how the same factor can really have these seemingly opposing effects at the same time. So we became interested in really understanding type 1 interferon and both the beneficial, so type 1 interferons are critical for the antiviral effects or antiviral effects during a virus infection. Without them, you really have the host has very much trouble controlling these viral infections. Are critical for activating the immune response, for leading to differentiation of specific myeloid and T cell subsets. But at the same time, it's hard to say chronic or too much because it's actually unclear, but at the same time, these same interferons induce this feedback inhibition that leads to the same loss of kind of T cell effector function, induces this immune exhaustion, can lead to actually resistance of the same therapies that are dependent on type 1 interferons. So how can these really diverse and large pathways induced by type 1 interferons lead in either of these directions? And of course, it's not either of these directions. It's a mix of both of these directions very much at the same time. But how can one of these weigh heavier on the immune response than the other? And so one of the first observations that got us interested in this area was made some time ago by a postdoc at the time, Liz Wilson, in the lab. And she noted that during these chronic infections, the expression of multiple interferon stimulated genes, so these are genes that are stimulated when cells encounter type 1 interferons, are actually sustained during chronic infection, whereas as expected, they're resolved with the resolution of virus and acute infection. These are sustained, and ultimately, despite not being able to really measure type 1 interferons themselves, interferon alpha beta, are present at very high levels in these cells. So what are these doing, we asked. And one of the first observations we made was that either using an interferon receptor knockout, so type 1 interferon receptor knockout, or an interferon receptor blocking antibodies, we found that the normally high expression of suppressive factors like IL-10, Pdl1 in these chronic infections are actually decreased in the absence of interferon signaling. And this was despite the fact that virus titers, so the antiviral effects of interferon being abrogated, led to higher virus titers. The fact that you saw that these negative regulatory factors were actually decreased despite having this increased virus replication. This was really the first time that we had seen this disconnect between levels of virus replication and the induction of these suppressive factors. They usually travel very much in tandem. And when we looked across the immune system, we noted that there was actually an increase almost across the boards of all immune subsets when interferon receptor signaling was inhibited. So you got a decrease of the suppressive and actually an increase of the inflammatory and immune increase when what would seemingly be necessary for this was blocked. Oh, that's not good. Let's see. Okay. Too many buttons. So in case you missed it, here's a quick recap of where we are now. There we go. And so with that, what we observed, and I think this was completely at least unexpected to me was that if you gave interferon receptor blocking antibodies for a defined amount of time, about a week, either at the onset of infection or during the middle of the chronic infection, and then broke this holiday and allowed interferon signaling to resume, we actually observed that this had the long-term effect of leading to decreased virus replication. So counterintuitive to kind of what we had thought or what we would have expected, if you provide a short interferon holiday, if you will, you actually provided the ability of the immune response to now have long-term control over this chronic LCMV infection. And we could find that this was dependent on CD4 cells, CD8 T cells, interferon gamma. So there were a variety of factors that came into play to allow this, but it revolved around this ability to kind of give this break in interferon signaling. And so kind of the aha moment or the thought for us was we had the actual ability to shift back from this suppressive state of kind of interferon signaling and reorient the immune response to more the beneficial side and allow these interferons to have more of some of these pro-immune qualities. So we wanted to ask, well, is this true in other systems or other chronic viral infections? And we wanted to ask this in a model in which we could modulate interferon signaling. So in collaboration with Scott Kitchen at UCLA, we set up a model in humanized NSG-BLT mice that were then, so these have a humanized immune system. These were infected with HIV. We then treated for a week, starting at about 10 or so weeks after infection with either isotype or anti-interferon receptor blocking antibodies. So HIV is, again, a disease that's characterized by this chronic type 1 interferon signaling. So if we blocked it, could we have this same beneficial effect as we saw in LCMV? And what we observed was that, in fact, blocking interferon for a week and then looking a little bit later at the CD8 T cells, we saw a decrease with interferon receptor signaling, so comparing black to red, in both the percent positive as well as the single cell level expression of multiple of these receptors associated with T cell exhaustion as well as with chronic immune activation. So blocking interferon allowed these to decrease these levels of immune activation on the CD8 T cells, and in fact increased the ability of these HIV-specific CD8 T cells to produce interferon gamma. So you saw a decrease in the overall levels of CD8 T cell exhaustion markers and an increase in their functional capacity that was then associated with a decrease in the ability of, sorry, I gotta move the pictures here, with a decrease in HIV titers. And this is comparing anti-interferon receptor blocked over isotype. So about a 5-fold decrease in HIV titers after interferon was blocked. And so, and when this was done in conjunction with antiretroviral therapy in these mice, this had an even much bigger effect. So by blocking interferon, we could decrease ISG expression, which I didn't have a chance to show. We decrease the overall levels of immune activation on CD8 T cells as well as CD4 T cells. We increased the function of HIV-specific CD8 T cells and ultimately decreased HIV titers in these mice. So again, like LCMV, this chronic HIV seemed to be driving this immune activation, and chronic interferon seemed to be driving this immune activation in HIV infection as well. So with this in mind, I moved to Toronto and I started in a cancer center, and I became very interested in trying to extend some of this stuff that we were doing in the chronic virus infection to wonder about whether these same mechanisms were shared in anti-cancer immunity. So in another situation where you had prolonged antigen stimulation, was this same kind of inflammatory suppressive balance being instilled, and was it regulating the immune system in the same way? And interferons have been shown certainly to be involved in almost every level of cancer establishment and progression in very different ways, for sure, but really being a player in every aspect of kind of the hallmarks of cancer, from both the initiation through to progression and ultimately through to metastasis. So we started wondering, well, what is their involvement? And then it's since then become clear that type 1 interferons are also critical for the positive and really the efficacy of enhanced immune responses to radiation therapy and immunotherapies, but also are involved in the feedback inhibition that causes resistance to these therapies. So we wanted to try and understand this complexity and diversity of really interferon signaling in human responses. And we've done a variety of projects, or we have a variety of projects ongoing, but one of the ones that I want to talk about today is what Giselle Bougalet in our lab has started to show is really a critical response of interferon, or at least an association with immunotherapy. And we hypothesized really that quantifying interferon stimulated genes, expression in patients undergoing cancer therapy, and in this case, anti-PD-1 based immunotherapies, would enable a better understanding of therapeutic outcomes. And we're by no stretch of the imagination the first people to have kind of that idea, but we thought that perhaps that more than simply on-off or a bulk analysis of how all cells are responding, that instead really understanding the single cell expression patterns of interferon reactivity would provide the key insights into the cells that are responding to therapy and how. So in essence, we thought that type 1 interferons may serve as an umbrella affecting different subsets of cells and cells within those subsets differently. So one cell type may have a negative response, may induce suppressive ISGs, whereas another cell type may respond very differently, may respond in a very positive way, whereas other cell types may have no response, and that ultimately it would be the integration of all these that would really provide the insight into how the immune system is sensing, understanding, and reacting to type 1 interferons. And so we developed a CyTOF panel to quantify multiple of these human interferon-stimulated proteins, ISPs in this case, across the immune system. And you'll have to forgive me because I'll use interferon-stimulated genes and interferon-stimulated proteins somewhat interchangeably, but they're not. And when I'm talking about them here for the remainder of this part of the talk, I'll be referring to interferon-stimulated proteins in this CyTOF panel. And this panel was comprised of both activating interferon-stimulated proteins such as MX1, PKR, as well as suppressive interferon proteins such as IL-10, Pdl1, IDO. Right, so we looked, and what we did is we looked in human PBMC, and this is a patient with melanoma, and this is a sample prior to anti-PD-1 therapy. And what we found is that across the immune system, different subsets of immune cells definitely had interferon signaling protein signatures that could define that cell subset, and these were different between different cells. But when we looked, although there were some very interesting actually differences in patients that ultimately would respond to therapy and those that would not, we didn't really have this robust response. It's in many ways, the patients that would and would not respond to therapy had similar basal levels of interferons signaling. And that's an oversimplification because there were interesting differences to tease apart. But one of the things that this made us think about was perhaps it's not how the PBMC, or these immune subsets, were responding to interferons prior to therapy, anti-PD-1 therapy that was going to be indicative, but rather how they will respond to these interferons that is ultimately going to be important for outcome or associated with outcome. So to test this, we set up a system in which patient PBMC were stimulated or were cultured either unstimulated or stimulated with interferon beta for 16 hours. And then, as you expect, we saw that using this CyTOF panel, we saw that there was an upregulation of these ISPs by when cultured with interferon beta, pretty much across the entire immune system, with some cell types showing a more enhanced reactivity for sure, but definitely a increase overall of interferon stimulated proteins with the culturing with interferon beta. And so Giselle developed a score, an interferon sensitivity score, that really took into account the average change in these individual ISPs between interferon stimulated and unstimulated conditions, and measured these across across all ISPs that we use. And so therefore we were able to generate really an average expression change in these subsets, where a higher score indicates more ISPs are expressed to a higher degree. So if just one ISP is expressed at a very high degree, since it will be divided by by the total ISPs at the end, this would be a less potent ISS score than if 3, 4, for example, ISPs are increased. And when we did this, what she saw is that the patients, and we focused here on the effector CD4 and CD8 T cell responses, although everything responds, these were the ones that responded most consistently. And what we found was that patients really stratified into either those that responded high or those that responded lower to interferon stimulation based on this ISS score. And we predicted, based on previous data from others, that those who would respond higher to interferon signaling actually would have a better outcome to anti-PD-1 therapy. However, like everything that's been going on in this interferon experiments, at least as far as I'm involved, this was absolutely not the case. Instead, what we observed was patients that responded with a lower overall interferon sensitivity, so a lower induction of interferon-induced proteins prior to therapy, ultimately had a better long-term survival, and this is CD4s and CD8s, and progression-free survival than patients who responded with a robust, a high interferon sensitivity. This was true in a second cohort of patients with melanoma. Again, this is looking, these are high and low designations determined prior to therapy. You see those that responded with a lower interferon sensitivity had a longer overall survival. And we then extended this into a small cohort of lung cancer patients. And again, we were able to see that those who responded with low interferon sensitivity had a much better overall survival than those who initially reacted with very high interferon sensitivity. So again, counterproductive, and so we wanted, or counterintuitive. So we wanted to try to understand, how does this relate to what's happening in the tumor? And so we looked at a well-established predictive 18-gene inflammatory signature that was published by Ayers et al. And that was indicative in tumors, was indicative of a high response, indicative of better responses to PD-1 therapy, whereas a low inflammatory signature was not. And what we observed was that unlike those responses in the tumor, the induction of interferon, the high and low induction of interferon, really did not relate to the inherent state of interferon or inflammatory signaling in the tumor. So there was really a disconnect between how these cells could respond in the periphery and what was actually happening within the tumor. However, one thing that did become obvious was that when we looked at these same patients after therapy, so 3 to 6 weeks after therapy, this is a treatment that's given every 3 weeks. So this would be after the first or second dose of this anti-PD-1 therapy. What we noted was that patients who had initially responded with a low interferon sensitivity actually increased their ability to respond on therapy, whereas those that initially responded very robustly actually had a diminished ability to respond, suggesting that those patients whose cells responded very hard and very fast actually did not maintain that ability to, at least in the periphery, to react to interferons with the same kind of inflammatory increase. And so we wondered, is this attenuated responsiveness on treatment due to the subsequent increased upregulation of suppressive ISPs, such as Pdl1, that then feedback to inhibit these subsequent interferon-induced responses? However, this was not the case, and we found that, although we found that interferons, that Pdl1 did in fact track very much with the rest of these interferon-stimulated genes with regards to induction, IDO, so endoleamine 2,3 deoxygenase 1 gene in this case, or protein, did not. And in fact, there was quite the disconnect between Pdl1 and IDO turned out to be very much correlated to treatment outcome. So there was this real kind of dissociation between how some of these suppressive ISGs or ISPs were reacting to treatment and it seemed to interferon induction. And it seems that some of these can actually have very much informative information for how these patients will ultimately respond. And so Giselle established, or is starting to establish, a model in which she could try and form a cutoff or some way to predict a priori how these patients will subsequently respond to an anti-PD-1 based immunotherapy based on the integration of multiple of these features that I've been talking about. And what she observed is in this model that patients that were predicted to respond, that were predicted to be at low risk, in fact, had a much higher proportion or a much higher likelihood of overall survival compared to those that were predicted to be at high risk based on the accumulation of these factors. And when we divided this up into high and low quartiles, this became even more apparent, and particularly for those patients who were not going to respond to therapy. And I think that predicting patients who will is, of course, a huge advantage, but I think that predicting those that will not is also infinitely important for ultimately guiding some of these therapies and trying to decide how to move forth with treatment. And so in conclusion for this part of the talk, we were able to show that how distinct PBMC subsets respond to interferon can be associated with outcomes following anti-PD-1 blockade. And this is PBMC in the peripheral blood mononuclear cells, and that the low responsiveness of effector CD4 and CD8 T cell subsets to interferon stimulation is actually associated with longer overall survival and progression-free survival in 3 different cancer types that we tested, and that the low interferon sensitivity is associated with the ability to maintain or perhaps even enhance these peripheral interferon responses. And taking all this into consideration, we can start to develop a model that perhaps could predict who will or who will not benefit from an anti-PD-1 based immunotherapy. And we also are starting to think that this might be a useful way to start to stratify patients for selection into either clinical trials or for arms in clinical trials. And we're starting to think about the utility of using this platform now for that kind of clinical trial stratification. We're currently working to uncover some of these mechanisms of why a patient would or would not respond robustly to interferons, and how this could be related either mechanistically or not mechanistically to outcome. And so really getting back to, how is this happening? One of the main questions we've really been trying to determine is, how does how are these opposing outcomes of type 1 interferon signaling chosen? Really, how do cells translate the inflammatory environment into the activating or suppressive interferon programs? And so with this in mind, we've started to identify factors that can function as feedback inhibitors to regulate the immune response in response to increasing amounts of interferon, and reciprocally tune down ISG expression and program T cell exhaustion. And at least for some of these, going in and changing their expression can actually reverse this T cell exhaustion and ultimately lead to much better cancer control. So we're starting to now play with some of that, and ultimately that will have to be for another talk, but I think is going to be a really exciting kind of set of experiments and data to follow. And so now to switch gears some, and I'd like to say that I'm switching gears, and I'd like to have some lead in, but it's just flat changing gears. What I'd like to talk about is another really kind of focus of our lab, which is how CD4 T cells are responding, regulated, and ultimately affected by immunotherapy in kind of these times of chronic antigen stimulation. And so we've been long interested in CD4 T cell help and trying to figure out what is the basis of really how do CD4 T cells guide the immune response. And it's well known that if you don't have CD4 T cells, that a variety of parameters of the immune system are really debilitated. But the factors that are directly driving that are in many cases not known. Certainly we know many, but there really seems to be a black box in understanding how we get from CD4 T cell differentiation to help of CD8 T cells, to help of dendritic cells or other myeloid cells, probably better known for B cells. And then ultimately, how do CD4 T cells drive their own differentiation and, for example, become cytolytic CD4s, able to kill virus infected or tumor cells? What are the mechanisms that lead to these divergent kind of immune outcomes, and how are they being guided? And a lot of this complexity has come from the fact that CD4 T cells are not just one homogeneous group in any given infection or any given state, but rather exist in a variety of different helper subsets that are characterized by transcriptional regulation, characterized by cytokine production, that ultimately then lead to kind of their focus, stimulating CD8 T cells for an antiviral response, stimulating B cells for an antiviral or antibacterial response, regulatory cells whose job is to tune down the immune system. So how are these being regulated in these infections? is of interest, but also trying to understand this is clouded by a lot of the complexity of having to do this on the backbone of cells with very different personalities. And of course, we show these in distinct branches, but there's also a great degree of plasticity and very much shared mechanisms of differentiation and effector function between many of these T helper subsets. So in virus infections, or at least for certain in chronic LCMV infection, what you see is really the predominance of virus-specific CD4 T cells differentiating into Th1 helper cells, so CD8 T cell sustaining, or T follicular helper cells, really these B cell sustaining CD4 T cells. And this is illustrated here. If you look at virus-specific CD4 T cells, 8 days into a chronic LCMV infection, you see that these divide into Th1 cells and TFH cells. However, by 30 days after infection, what you see is there's a real loss in these virus-specific CD4 Th1 cells and a real accumulation of these TFH cells. And this is due to both attrition of these Th1 cells as well as a transformation of them into these TFH cells. And it's not to say that having TFH cells is bad. It's certainly not. You absolutely need them. But like most things in life, we started to think that perhaps a balanced response would be even more beneficial. So we wondered, well, can we restore this Th1 response by adding new virus-specific, so naive virus-specific CD4 T cells into the middle of the infection? And would they then differentiate much like this, generating Th1 and TFH cells? But to our surprise, what we found instead was that these late prime cells actually almost exclusively generated TFH cells with very few Th1 cells. So really there was this ongoing and new restriction on the ability to maintain CD4 Th1, virus-specific CD4 Th1 cells during this chronic infection. And we went on to show that this was due to type 1 interferon changing dendritic cells from these stimulatory to these more suppressive phenotypes expressing IL-10 and Pdl1, which together inhibited Th1 differentiation while promoting TFH differentiation. So really the environment was set up to inhibit Th1 cells. So we asked, well, why? What would be the impact then of introducing Th1 cells into this environment? So we in vitro primed virus-specific CD4 T cells into Th1 cells, or we transferred, again, these late prime cells so that they would be TFH cells. And then we asked, how does the introduction of these TFH or Th1 cells affect the immune response? And what we observed was that when we added the Th1 cells, we could prevent this normal decay of the virus-specific CD8 T cell response that happens over the course of the chronic infection. That in addition to the increased number, we could increase the frequency of increase the amount of these functional virus-specific CD8 T cells. You could see when the Th1 cells were present, the CD8s now had a better long-term ability. So again, this is at this or around this time point here. These cells had a sustained function, and ultimately, there was an enhanced control of LCMV and chronic LCMV infection when these Th1 cells were introduced. So this said to us that this lack of the loss of these Th1 cells and the lack of the ability to generate these new Th1 cells could have a pathogenic effect on CD8 T cells and virus control. And ultimately, we began to think that many of the CD8 T cell dysfunctions observed in chronic virus infection may be due to a loss of this CD4 Th1 help as the infection progresses. So this got us into thinking, well, this is fine and interesting to transfer these Th1 cells, but could we restore the function at this point of any of these? Could we bring out any of these responses? And in particular, could we perhaps bring out these Th1 responses by blocking some of the inhibitory pathways that are really present and bogging down the immune response in these chronic infections? And so we first focused on the Pdl1 pathway because of its obvious role and well-known role in in the clinic in terms of restoring immune function and enhancing CD8 T cells, enhancing control of cancer. So we wanted to ask, well, what is this doing to the CD4 T cells? And really this stemmed from the fact that it's not exactly clear how PD1 or Pdl1 affects antigen-specific CD4 T cells. The general thought from many cancer models is that they are not really functionally restored in vivo by blocking PD1. However, in the clinic, in many kind of clinical settings, what's seen that PDL treatment does in fact associate with increased CD4 T cells. And in fact, blocking PDL1 can lead to control of beta-2 microglobulin, so MHC1 deficient tumors like Hodgkin's lymphomas. And this has been associated with CD4 T cell responses. So it seemed that this PD1, PDL1 was in fact affecting the CD4 T cells, but how it was doing was really unclear. And we hypothesized that perhaps it wasn't that these PD1, PDL1 interactions are blanketly just taking out or suppressing the CD4 T cells, but that perhaps they're doing this in a very subset specific manner, and that by interrupting these signals, one could enhance distinct subsets of CD4 Th cells. And so we instituted a system in chronic LCMV where we blocked PDL1 beginning at day 25 after the initiation of chronic infection, then blocked every 3 days, and then analyzed the virus-specific CD4 response either after the first treatment or after the third treatment. And using a CyTOF panel that could really nicely dissect various aspects or various subsets of CD4 Th cells, we observed that there was, in fact, in response to anti-PDL1, a change both in abundance as well as in number of a subset of clusters that increased actually with the anti-PDL1 therapy, right? So not everything did, but there was a subset of cells that seemed to increase when PDL1 was blocked. And when we looked deeper at these, what we observed was that all the cells expressed PD1. So it wasn't just that certain cells expressed PD1 and others did not, but rather that the subset that was enhanced specifically was in this TCF negative group. So for these CD4 T cells, this would be associated with a non-TCF being associated with a TFH type cell. And you see these bona fide TFH, this BCL6 bright, CD69 bright population, but rather an enhancement in a population of cells that was TIM3 positive, really CD39 positive, TBAT positive, SLAM positive. So really this Th1, this small population of Th1 cells that were present within, that remained within this virus-specific CD4 T cell group, seemed to be the subsets that were being enhanced when PDL1 was blocked. And one of the things that also really stood out that I thought was really cool was that it's not, again, just Th1, TFH, but rather that you have real heterogeneity among the response. So you have this whole group of cells that expresses markers associated with Th1, but also associated with TFH, suggesting that there are really these kind of Th1, TFH hybrid cells that exist in these chronic infections. And how they're kind of seeding each other is of course something that will be of interest moving forth. But in addition to the increase in these Th1 cells, we saw an increase in Th1 cytokines. So for example, interferon gamma, both an increase in the number as well as the single cell expression of interferon gamma, and an increase in IL-10, which would be consistent with feedback inhibition, as well as also there's a nice set of data out that suggests that IL-10 is really expressed by Th1 cells. So again, this idea that you're blocking PD1 or PDL1, and this is allowing for an amplification of the Th1 cells, as well as an increase in their cytokine producing capacity. And so we asked, well, why is this subset, this small subset, being selectively increased? What's allowing for this? They all express PD1. And one of the observations we made was that the was that the cells that were ultimately amplified by, or the subsets that were amplified following anti-PDL1 therapy, were those that were already in cycle. And this is based on Ki67 staining. And importantly, these were not clustered with Ki67. So this is how Ki67 looks in these otherwise clustered subsets, suggesting that really PDL1 blockade induced cell clusters that were already in cycle as opposed to inducing new cell populations into cycle. And this was something we also observed for virus-specific CD8 T cells. So if that was the case, we wondered, well, where were these cells then coming from within? How was the path of these cells being affected by this PDL1 blockade? And what Laura Snell in the lab did, and sorry, this is all her work. I forgot to point that out. What she did is she did a pseudotime trajectory analysis based on this CyTOF data. She then overlaid onto that the clusters from the CyTOF, so the established clusters. And what she observed was that the pseudotime algorithm defined 3 tips, or 3 really endpoints or beginning points in this data. One in the most highly differentiated cluster 8, one in the TFH cluster 2, highly differentiated TFH, and one in this more intermediate cluster 6 here. She then overlaid the increase in numbers. So what clusters are actually increasing numerically when PDL1 is blocked, and was able to show based on this analysis that really what was happening was that this cluster 6 was being pushed with anti-PDL1 blockade much towards this differentiated cluster 8. So you were driving these cells through this kind of a differentiation when PDL1 was blocked. And this was interesting because to us, this really suggested also that you were stopping or impeding this normal differentiation of these TH1-like cells down this TFH path, like I had showed previously. So you were redirecting the way that these cells were differentiating during this chronic infection when you interrupted this PDL1-PD1 axis. So I think what was also interesting was when we looked at Treg cells, we observed that similar to the virus-specific CD4 T cells, these Tregs were also activated, and we were not certainly the first to show this, but what we observed was that these were the TCF negative population of these Tregs were in fact the ones that were really increasing with abundance. And when we looked, what we observed was that similar to the virus-specific CD4 T cells, these consisted of really these TH1 clusters of Treg cells and these TCF negative clusters. So again, it appears that PDL1 blockade was able to amplify both the CD4 virus-specific TH1 cells as well as these TH1 phenotype Treg cells. And we were able to show that in fact, this Treg depletion increased the CD4 TH1 effector cells, but not TFH cells. So really, this PD1 block or PD1's signaling or suppression by these CD4 T cells is really dampening these TH1 responses in these chronic infections. And this appears to be system-wide for both the virus-specific response as well as the Treg response. And so we asked, well, why isn't this what's observed in these cancer models? What's going on? And certainly when we looked in a breast tumor model, and this is a breast tumor model engineered so that we could identify tumor-specific cells based on an LCMB epitope. We observed that there were certainly TH1 phenotype cells within the tumor. This is based on TBET expression here, although we didn't confirm that with other markers. However, when we looked more closely, what we found is that really these TH1 cells were largely in this Treg population, but when we looked at the tumor-specific cells, they hadn't really differentiated into this Th1 phenotype. So really what was happening was the PD-1 was having an effect. We do see amplification of the Treg cells, but the tumor-specific cells, at least in this model and an MC38 model, were not of a subset that was capable or being targeted by this interruption of these PD-1, Pdl1 signals. So that started to explain to us perhaps why we were not observing this effect or this effect of blocking PD-1 on the tumor-specific CD4 T cells. And so we next asked, well, we tried to get a deeper understanding of why these Th1 cells are specifically being amplified. And so we did single cell RNA-seq on these virus-specific CD4 T cells after Pdl1 treatment or an isotype treatment. And again, we saw an amplification of these clusters here, which corresponded to Th1 cells. So this was nice because it validated a lot of what we'd done in Cytoph. And we were able to gain a lot of insight, but I want to wind down this talk by showing one of the, I think, more interesting things that we were able to observe was that when we looked at these Th1 cluster 2 and cluster 3 in anti-Pdl1 versus isotype treated, we saw an increase in pathways that were associated with effector CD8 T cells. So again, these are CD4s, but associated with effector CD8 T cells and of leukocyte-mediated cytotoxicity. And so when we look deeper, we in fact saw that many cytotoxic mediators were increased in these clusters 2 and 3 in anti-Pdl1 treated condition versus isotype. So really, the anti-Pdl1 was turning on a cytotoxic signature in these virus-specific CD4 T cells. And so we wanted to ask, well, is this meaningful? Is this actually resulting in enhanced CTL capacity of these virus-specific CD4 T cells? So we performed an in vivo CD4 CTL killing assay in which splenocytes from MHC1 deficient mice, so these mice lack beta-2 microglobulin, so can't be targeted by CD8 T cells, were isolated. These include both MHC1 and MHC2 negative cells, but all MHC2 positive or MHC2 negative cells, but all MHC1 negative. These were divided in half and labeled with a peptide that would be identified either by our LCMV specific CD4 T cells, or in this case, an irrelevant peptide, ovalbumin peptide that would not be identified, would serve as background killing. These were combined and then transferred into chronic LCMV infected mice that had been treated with anti-Pdl1 blocking antibodies. And happily, what we observed was that MHC2 deficient cells within this group, so for example, CD4 cells or CD8 cells in this group, were not targeted, were not lysed. You saw a similar amount or frequency of LCMV labeled and OVA labeled targets in naive or LCMV infected animals. So it was not background killing that is seemed to be occurring here. When we looked at MHC2 positive targets, so really targets of CD4s, what we observed was that, as has been shown before, that if we injected these into mice infected with acute LCMV infection, you got very strong CTL, CD4 CTL activity, killing about 55–60% specific killing. However, when we looked in chronic LCMV, the CD4 CTL capacity was almost entirely abolished. And when we blocked Pdl1, you now got an almost complete restoration of the ability of these CD4 T cells to kill their targets. So chronic LCMV seemed to completely abolish the CTL activity, which was then restored when Pdl1 was blocked. So kind of to conclude this part in this talk, we were able to show that Pdl1 targets, or Pdl1 targets virus-specific CD4 T cells and Tregs, but does so in a very subset-specific fashion despite system-wide PD-1 expression. And Pdl1 specifically restricts CD4 Th1 cell amplification, cytokine production, and suppresses CD4 CTL killing capacity during the chronic infection, and that the lack of Th1 tumor-specific cells, or maybe other susceptible cell Th subsets in various cancer models, may account for why PD-1, Pdl1 blockade has not been associated with really functional restoration of CD4 T cells. And importantly, blocking Pdl1 could amplify these virus-specific CD4 T cells with minimal effect on the much more predominant virus-specific CD4 TFH cells. This, I didn't have a chance to show this, but this is cell intrinsic, as we see a very similar outcome when we use PD-1 deficient virus-specific CD4 T cells. And Pdl1 blockade is able to target Th1 phenotype Treg and specifically amplify existing populations of these cells that appear to already be in cycle. So target these Th1 cells, perhaps largely because they're already in cycle and being restrained by this PD-1, and rapidly turn on the ability of these Th1 cells to produce cytokines and reprogram their cytolytic capacity to now be able to kill target cells. And I just want to note that it's important to say that we show here this is true for Th1 cells. However, in other models where other CD4 Th subsets are proliferative or are in cycle, these too may be targets of anti-Pdl1. So really it comes down to largely probably the receptive state of the Th subsets that dictates how these will ultimately respond, or their ability to respond to this therapy. And so with that, I'd like to thank everybody who has made this possible, and everybody in this slide. It's a group effort, honestly, but to point out a few who really contributed to the data I showed. Heidi, who's involved in everything in this lab. Giselle, whose work was on the human, the interferon signature in the human, responsiveness to anti-PD-1 therapy. I'd like to thank again Laura Snell, who's done really all of this CD4 work, who just recently left to start her lab in at the University of Indiana. Mangdi, who's working on CD4 T cells in cancer system, some of that data I showed, as well as many people at Princess Margaret and University of Toronto who've contributed, including Tracy, who's helped with a lot of the single cell data, Marcus and Adrian and Trevor, who've been critical for a lot of the clinical aspects of this study, as well as our whole immune profiling team, Ben and Diana, and then those who are critical for the HIV and our TB work, including Scott, Angie, and Jerry. So with that, I'd like to thank everybody, and thank you. I look forward to Twitter questions. This will be my first time as a Twitter question answerer. So thank you.
Thank you so much, David. It was a wonderful talk. Thank you. Very, very precious, the predictive value of your research in the first part, and very intriguing, the effect of PD-1 on the cytolytic capacity of CD4 T cells and all the rest. Thank you so much for sharing your research, published and unpublished, here in Global Immunotalks. So just in case if somebody missed it at the beginning, I would like to quickly remind people how to ask questions via Twitter. And so let me project this slide here. And so if you'd like to ask questions for David, please search the account Global Immunotalks, find a tweet that says ask questions for Dr. David Brook here, and reply to that tweet with the hashtag Global Immuno, and David will use the Brooks Lab handle to answer the questions. And I see, David, that you already have a question there, so I hope people that are watching also in YouTube will ask questions and take the opportunity to interact with David in Twitter. Thank you so much, David, and everyone for joining today. And I hope to connect with you again next Wednesday. Okay, take care.
Thank you. Thanks.
Bye. Take care, Dave. Thank you.